dear colleagues, dear friends. It's a pleasure to give this talk. To be honest, I was a bit nervous because I said, what I'm going to talk about here, because there's literally nothing published in that regard. So I thought I'm going to give you actually an intro on what is trained immunity and where could it lead into the future. And in the very end, I'll show you some novel data which are yet unpublished, but might really change how we treat psoriasis going forward, not just psoriasis, but a lot of other inflammatory diseases. So just to start with some really textbook knowledge, going back to when I was a med student, and then I'll start criticizing that or questioning that a little bit going forward and showing what has actually happened over the last 15 years or so. So we know the skin, and this I don't have to tell you, is probably the most exposed surface that we have, and one of these hazards is obviously the microbiota, and we have more bacteria on us than actually we have cells, human cells. We have more than almost 2 kilos of bacteria on us, 600 grams on the skin, so this is a huge amount. And we have to mount an immune response against that. And the skin is obviously the first line of defense. It's a physical response in one regard. It has to feel when it is breached, but it's at the same time also the first line of immune defense. So the keratinocytes produce antimicrobial peptides, they produce the complement system which are secreted, and then you have an immune response which immediately contains the infection, immediately contains and stops it, or at least slows it down, so we can actually mount a direct immune response or an adaptive immune response, which then is antigen specific. So the question is, how do they sense it? Because they do not have any specific antigen-specific receptors. So they do that through the so-called PRRs, so the pattern recognition receptors, so they distinguish between self and non-self through these so-called PAMs. These are pathogen-associated molecular patterns, which are highly conserved structures on the surface of bacteria or pathogens in general, which are essential for the survival of these pathogens. So they do identify actually a whole family of pathogens non-specifically. And these PRRs include obviously toll-like receptors, but also not-like receptors, AIM2-like receptors, C-type lectin and RIG-like receptors, and they're expressed on the surface of the cells in endosomes, but also in the cytoplasm, depending on where they need to actually sense those PAMs. And this can also be in the context of sterile inflammation upon tissue damage, be a damp, so-called damage-associated molecular patterns. It can also in a sterile infection, sterile inflammation can be induced. So this is a very rapid process which is non-specific and leads to phagocytosis, recruitment of other cells or wound healing as well, but then obviously through particularly cytokine production also to the response, to the adaptive immune response with cells which are then recruited into the cell as well. And this is a rather new concept, or not new concept, but new evolutionary process. It's only about 500 million years old, is the adaptive immune system, which is obviously highly antigen specific, includes T and B cells, is slower, so it takes days, while the other one takes minutes to hours. When it leaves then these highly differentiated immune cells, in memory cells, which are even subtypes of cells, which Th1, Th17, and so on. So this is a bit part of what I wanted to say about what is textbook knowledge from 30 years ago when I was in med school. I'm getting old. And so this part in here on the left it's a primitive response, which is all the cells or all the organism have, while on the right, as mentioned, it's only invertebrates, and this is only about 500 million years old. And to the left, the innate immunity does not have an immunological memory, while to the right, there's a persisting immunological memory, which is highly specific. And this is a huge advantage for survival. And if you think about it, that doesn't make any sense. And now it's the changing of the paradigm, because we actually only represent about less than 1% of the biomass on Earth is the vertebrates. If you look at biodiversity, it's in the range of 3 to 5%. So the non-vertebrates have a huge success on the Earth, and they would not have any memory. So this doesn't make any sense at all. So you need some memory, and you need some defense against a recurrent infection. And this actually now we know is true, so there is innate immunity, and that's what is called actually trained immunity. And so you can have an infection or live vaccine, for example, BCG vaccine, which leads to protection not only against a specific pathogen, but also against others. For example, if you do BCG vaccinations, this leads to protection against lethal candidiasis, even in SCID mice, which lack T and B cells. So there is a cross-protection that you have in a subsequent response, which then is much higher. And this is innate immunity, or as mentioned before, this is trained immunity. And how does this work? And this is work done over the last 15 years. So there is epigenetic reprogramming in the cells, in contrast to real genetic recombination in the adaptive immune system. Here you have epigenetic reprogramming. These are histone modifications, DNA methylations, long non-coding RNA, which is expressed, and modulation through microRNA. And this all leads actually to that the chromatin section, particularly at given sites, at pro-inflammatory genes, are more accessible. And so in the left here, you can see it in a steady state, as mentioned before. I think it does work. So you have in a steady state highly condensed chromatin. It is not accessible, so you cannot have transcription, and have a high DNA methylation. Then when these cells becomes stimulated, you have the chromatin opens up. You have, if the cell undergoes training, some marks which are produced in there on specific sites that you have a pro-inflammation afterwards, but I'll get into that. And then they go back into a resting state. If the cell is not trained and there's no reprogramming, it goes back to the same as it has been before, so in a steady state. However, if it has been depending on the stimulus, it goes into a state where the chromatin is not completely condensed anymore, but remains partially open, particularly at this pro-inflammatory site, which then you use, for example, IL-6 or TNF or others. And then you have a mild DNA methylation, so it is not the same anymore, so you can detect that through things like Johan just said before, like ATAC-seq. And then if you have a re-stimulation, the response is much faster, it's much stronger, and it can qualitatively also be different. So in the end, what you have is a retained innate immunity on a cellular level through an accessibility of the chromatin, which leads to this long-lasting responsiveness of the cells, which is changed. And this can go either way. So this can be trained. So you can have a cell which has a monocyte, so it is innate immune cells. You have an infection, you go on epigenetic modification. You have a trained cell, who in the second run then shows again a much stronger response down the road. But you can also have tolerization. So the cell is actually, the response is blunted then, and you have less immune response. And this means that the chromatin actually stays condensed, and you have no accessibility of these pro-inflammatory cells. And this obviously is to prevent an overshooting immune response and tissue damage. And this is something we know as LPS tolerance, for example. So after sepsis, you have immune paralysis. Or what for us is more common, if you have recurrent erysipelas, the second, third, or fourth time, patients do not show fever, they do not show CRP increase, and this is explained through changes on the epigenetic level. So you can have actually two pathways. So you have an initial stimulus, the cell then goes back either to homeostasis and resting, so no training. It goes into this trained immunity where it is much more easily activated and you have a stronger and faster response, or it goes into the tolerance and have no or a weaker response down the road. So this is obviously a system which is preventing us for infections particularly, but it can also be in the case of inflammatory diseases. So under the evolutionary pressure, this was established, but if there's a misguided, aberrant inflammatory response afterwards, this can have a harmful effect. And this has been shown now very recently, for example, for long COVID, that you have epigenetic changes in these patients which have long COVID, which have a much easier inflammation on the slightest stimuli. On the other hand, it has been shown that, for example, Western type diet, Western type lifestyle changes actually induces epigenetic changes on monocytes. It leads to inflammation, and even if you stop this diet, they remain, and patients have hypercholesterolemia and then leading to atherosclerosis. And this has been shown in mice very nicely, but it also been found in patients with atherosclerosis. You do find these monocytes in these blood vessels that are epigenetically altered. And again, a bit closer to us, I mean, Joel, you're working on that, but most of us are rather in dermatology. So you can see that there's also something which we have a problem with for a very long time. How can it be that patients for living on a farms actually have much less asthma, allergic asthma or hay fever? And you can see that these patients actually that are undergone this contact with this farm dust, they have a protective epigenetic changes and changes the mucosal tolerance, and therefore can actually prevent from raising up to a more severe asthma. While on the other hand, in a more urban situation, more sterile situation, you have other contact which actually it leads to training, and this leads then to asthma or increased allergies. So this explains actually quite a bit of also these overshooting immune responses, which had been a problem in previous concepts. So, but there's one problem which still remains. I mean, if these cells are actually undergoing training or tolerance, they go into this state, and afterwards, these cells have a half-life of about 5 to 7 days. So any training that would happen in these cells stays on for 5 to 7 days. That's not really relevant. And this was a conundrum which was actually recently solved, because it was shown that you do have central trained memories, and hematopoietic stem cells can undergo a training upon vaccination, upon an infection, and they actually have these marks that I showed before, so this histone modification, which are actually translated to daughter cells and which then lead to peripheral trained immunity, which can stay on for months or even years. On top of that, what has also been shown very recently by Elaine Fuchs and Shruti Naik, you do also have epidermal stem cells which can actually undergo epigenetic changes, and you do have epidermal stem cell inflammatory memory, and this particularly on the sites where you have the most contact with pathogens, so the skin, the lung, and the intestines. And how does that work? That's actually a nice study, and now I'm getting closer to psoriasis, because that's the idea that I was asked from Hervé. Where does trained immunity lie in psoriasis? So what they did, they actually stimulated the mouse with imiquimod, so a psoriasis-like a model, and what you have inflammation, you have acanthosis, you have transcription of pro-inflammatory cytokines, and this subsides eventually. In clinically, histologically, but also transcriptionally, they're completely back to normal after 30 days. But what we do have is this memory domain. So they do have domains, we've shown before, that remain accessible, that remain open also in keratinocytes. And then 6 months after the first stimulus, a second stimulus, and this is not the same, this is innate immunity, you do not need the same stimulus, they did just wounding, and they had a much stronger response, which a much stronger inflammatory response and a faster wound healing. And this is shown, what they've shown is that this is particularly the AP1 family of transcription factor, phospho-Jun, which remain open and then can lead to this inflammation. I'll come back to that in a second. So what about psoriasis? and we do have disease memory, that's clear. Disease comes back, it's a recurrent disease, chronic obviously, and we know that the T cells are absolutely critical, that they are the driver of a recurrent flare, but we also do have an inflammatory memory, epigenetic changes, or are thought to have one, and this is what I wanted to comment into very end. While it is a long-standing memory, it is shorter than the specific one, the adaptive one, and it might be actually changed. Because what Lars Iverson recently showed nicely is that the disease does not come back randomly. It comes usually back at the very same spots. And this was for a long time explained that there's obviously disease memory within the skin, that these are the T cells. And this was Levitzmo, which showed that you have, upon healing of the lesions, you have the tissue-resident memory T cells which remain in there, which are locally reactivated, and the recruitment of T cells, circulating T cells, you have a clinical relapse. So if you want to modify the disease, if you want to go for cure, because Johan was talking about cure before, you would have to get rid of these skin-resident memory T cells. And this is probably extremely difficult, if not impossible, because the T cells, they protect us against infection. And if you would just deplete them by treating IL-23 blocker with IL-23 blockers, IL-17 blockers, that will mean within the long run, they would also remove the physiological tissue-resident memory T cells, which protect us against infection. And the one thing these therapies are not linked to or associated to is skin infections. So it's very unlikely, and all the data also speak to that we cannot remove the tissue-resident memory T cells. So can we modify the disease course, or again, as Chris was suggesting or challenging me a few years ago in Tokyo, do we talk about cure? Can we cure some patients? And if you look at the data, from the withdrawal data, with how long does it take the disease comes back, some data, and this is not just the one here from Novartis with secukinumab, but also all the others, short disease duration is a predictor that the disease does not come back so easily. And here you can actually see the most granular data yet published, and you can see the ones with a short disease duration. So just 1 year after the disease occurred, they were treated with secukinumab. The majority of patients still remain disease-free and relapse-free without any therapy. If you wait for 5 years, then it doesn't matter if the patient has a disease duration of 5 years or 15 years, they show the same chance of staying relapse-free without therapy. So something happens between 1 year and 5 years that you lose your chance, your increased chance to actually modify your disease. So early intervention seems critical in that regard. One option would be that we just hinder the T cells to actually migrate into the cell and populate the skin with these skin-resident memory T cells. But again, it's very unlikely that this would really alter the course, because if you wait for a year or two, you stop the therapy, then they would just start again migrating into the skin. So what Novartis then did, they based on the data I just showed before, they established or planned the step-in study. So they looked at new onset psoriasis, less than 1 year treatment duration, or the patient with long-standing disease more than 5 years, and treated those patients with secukinumab for 1 year. So first, we look at the baseline. So is there a difference between long-standing disease and new onset psoriasis? Clinically, histologically, and concerning the skin-resident memory T cells, they're absolutely identical. But if you look at the epigenetic changes, you can see that exactly those changes, among the changes which were different, but the most prominent ones were exactly AP1 family members, June and Foss, which Elaine Fuchs and Shruti Knight showed are critical for the recurrence of an inflammation, indicating that really keratinocytes are the ones, this was through deconvolution, we identified that these changes are on keratinocytes. This was prior to what Johan just showed, that we have single cell ataxic and spatial. This is just bulk data, but through deconvolution we could identify that those changes are actually on the keratinocytes. So it seems that exactly those memory domains on the keratinocytes are actually increasing the risk for recurrent disease, so indicating that actually the keratinocytes are orchestrating the recurrence of the disease. Now the question was, what happens after therapy? And if you, after 1 year of therapy, the clinic goes into remission. Histologically, they are identical. There's no difference between new onset and long-standing disease. And while it takes a bit longer for the gene expression to go down, you can see that after 1 year also the transcription is completely back to normal. So clinically, histologically, but also on the transcription level, those are not distinguishable. However, if you look at the epigenetic, there's a mistake here, but if you look at the epigenetic signature of those patients, you can see that the long-standing disease has an epigenetic score which is not reversible. So you cannot reverse completely the epigenetic changes if you wait for 5 years or longer, while in the new onset psoriasis, you can see that they're going back immediately, go back to normal. So this indicates if you want to change disease, if you want to change the disease course and maybe aim for cure, you would have to go early intervention, and you can still prevent or reverse the epigenetic changes in these patients. So back to the data. The rest I'm not allowed to show because it's not yet published, but you can see that these changes, so the AP1 family transcription factors, they are actually reversible. So it's not the ones which remain, it's other factors which play a role, also pro-inflammatory and also on the keratinocytes, but it is not the AP1 family domain. So in conclusion, trained immunity is a rather new concept, but absolutely critical, that protects us against infection, not us, but in general, against infections through this long-term functional epigenetic reprogramming. And this is obviously through epigenetic, for central memory and through the epithelial stem cell changes, that this can be a long-term protection or a long-term memory. And what is important is it's a bidirectional communication between the keratinocytes and the immune cells. So immune cells actually leads to a wound response, but on the same side, the keratinocytes can also induce immune response, and this is particularly important in psoriasis, as it seems like that really epigenetic changes in the keratinocytes make the patients prone to recurrency at the same site after remission. And this also speaks to the fact that with early intervention might prevent these changes and or at least can reverse these epigenetic changes, so we can modify the disease course of the patient and potentially in some patients lead to cure. Now the question in the future is early intervention enough? Can we, as it is a short-term memory, can we just treat the patients longer and then these changes will become reversible, or should we add some therapies that are actually targeting directly epigenetic changes? There's one called mTORs or sirolimus or rapamycin, IL-1 therapies, so that therapies that we combine with classical therapies to actually also alter these epigenetic changes. And this obviously has impact not just for psoriasis, but also other diseases such as alopecia areata, vitiligo, where we know that early intervention also can alter the disease course, or even more so HS, where again we heard about smoking, and smoking is one of the key drivers of epigenetic changes. And while we still tell every patient to stop smoking, it doesn't really change the disease course in HS. So it might be that we actually have to prevent the epigenetic changes from happening, and therefore we can do something in modifying the disease course. And with that, as I mentioned, there's very little data published, but I hope it instigated a little interest and new research in the field. Thanks a lot.
Exploratory topic, very nice way. I can see there are some questions. First, Andy Blowelt and then Joe Gellman.
So one of the constraints of studying early disease is entry criteria. And I think it's always been, 10% BSA, PASI 12, but the cat may be out of the bag if that continues to be the entry criteria for short disease duration. So I've been advocating that if we do short disease duration studies or guttate psoriasis studies, that we have to lower the severity criteria, because it may be too late if you keep the same high-level entry criteria. Do you feel that way, or?
Absolutely. I mean, we discussed a little bit in that regard in Madrid, but I completely agree. I think we should probably proactively treat the patient much earlier. And we cannot treat every single psoriasis patient with a biologic. That's not just doable, at least not everything outside of the US. But I think the entry point should be lower. We have to identify through biomarkers or prediction, more or less, if they evolve towards severe disease, and then just go for 1 year, maybe 2 years with the biologic, and then stop and see maybe we can actually cure 30% of the patients. Same with Goucher psoriasis, and it might be true, and we're doing a study, Chris and the SSF has launched that. It might be also there that there's actually epigenetic changes which predict that some patients go into these recurrent flares or do just one flare, or the ones which go into a chronic disease. This is still yet absolutely not known, but I completely agree. We should not wait for 5 years, do phototherapy, it's not really working, do a little bit of methotrexate, it's not working. And only 5, 6, 10 years down the road do something more aggressively. I mean, cheaper, we could do cyclosporin for half a year or something and see if that works. So it's not like that we need to have these probably very expensive targets.
That's a quick question. So when does psoriasis actually start? And the context of this, like in lupus, we know that patients have antibodies about 5 to 10 years before they present with clinical symptoms. Now, we have the advantage in dermatology, we can sort of see stuff, but really the question is, when do they actually start having disease, when you can see it?
Yeah, difficult question. I think for at least for the changes in, or epigenetic changes in the keratinocytes, you need inflammations. You need sufficient stimulus to train those cells and induce a persistent epigenetic changes. So I would assume it is a relevant inflammation. Again, if there's a trigger that we have very fast evolving, if that's a different population and they undergo more of these changes, difficult to say, because we do not compare exactly the same populations in this study.
So you don't think it's subclinical? You don't think people have smoldering?
I think for epigenetic change, at least on the skin, I doubt it. Yeah.